Tonight's lecture is over Chapter 3, the cellular level of organization, and this chapter begins on page 63 in your textbook. Cells carry out a multitude of functions that help each system contribute to the homeostasis of the entire body. At the same time, all cells share key structures and functions that support their intense activities. The purpose of this chapter is to introduce the parts of a cell, discuss the importance of the plasma membrane, discuss the components of the cytoplasm, compare and contrast mitosis and meiosis, and understand the effects of aging and what aging does to the cell. So moving on to the parts of the cell, the average adult human body consists of more than 100 trillion cells. Cells are the basic living structural and functional units of the body. The scientific study of cells is called cellular biology or cytology. The plasma membrane is the cell's flexible outer surface separating the cell's internal environment from the external environment. It is a selective barrier that regulates the flow of materials into and out of the cell. This selectivity helps establish and maintain the appropriate environment for normal cellular activities. The plasma membrane also plays a key role in communication among cells and between cells and their external environment. The cytoplasm consists of all the cellular contents between the plasma membrane and the nucleus. This compartment has two components, the cytosol and organelles. Cytosol is the fluid portion of the cytoplasm, and it's also called intracellular fluid. It contains water, dissolved solutes, and suspended particles. Within the cytosol are several different types of organelles. Each type of organelle has a characteristic shape and a specific function. Examples of organelles include the cytoskeleton, ribosomes, the endoplasmic reticulum, the Golgi apparatus, lysosomes, peroxisomes, and mitochondria. The nucleus is a large organelle that houses most of the cell's DNA. Within the nucleus, each chromosome, a single molecule of DNA associated with several proteins, contains thousands of hereditary units called genes that control most aspects of cellular structure and function. And that brings us to figure 3.1 on page 64. And this just shows a cartoon of a cell. So remember, the cell is the basic living structural and functional unit of the body. And this cartoon is figure 3.2 on page 65. The plasma membrane, a flexible yet sturdy barrier that surrounds and contains the cytoplasm of a cell, is best described by using a structural model called the fluid mosaic model. According to this model, the molecular arrangement of the plasma membrane resembles a continually moving sea of fluid lipids that contains a mosaic of many different proteins. Some proteins float freely, like icebergs, in the lipid sea, whereas others are anchored at specific locations like islands. The membrane lipids allow the passage of several types of lipid solid molecules, but act as a barrier to the entry or exit of charged or polar substances. Some of the proteins in the plasma membrane allow the movement of polar molecules and ions into the cell and out of the cell. Other proteins can act as signal receptors or as molecules that link the plasma membrane to intracellular or extracellular proteins. If you look in the upper right corner of this cartoon, you can see the basic structural framework of the plasma membrane, and that is the lipid bilayer. The lipid bilayer has two back-to-back layers made up of three types of lipid molecules: phospholipids, cholesterol, and glycolipids. About 75% of the membrane lipids are phospholipids. Phospholipids are lipids that contain phosphorus. Present in smaller amounts are cholesterol. Cholesterol is a steroid with an attached hydroxyl group. And there are also various glycolipids. These are lipids with attached carbohydrate groups. Moving on to membrane proteins. Membrane proteins are classified as integral or peripheral according to whether they are firmly embedded in the membrane or not. Integral proteins extend into or through the lipid bilayer and are firmly embedded in it. Most integral proteins are transmembrane proteins, which means they span the entire lipid bilayer and protrude into both the cytosol and extracellular fluid. However, a few integral proteins are tightly attached to one side of the bilayer by covalent bonding to fatty acids. Peripheral proteins are not as firmly embedded into the membrane. They are attached to the polar heads of the membrane lipids or to integral proteins at the inner or outer surface of the membrane. And this slide is figure 3.3 on page 67. Membrane proteins largely reflect the functions a cell can perform. For instance, an ion channel, which is an integral protein, forms a pore through which a specific ion can flow to get across the plasma membrane. Most plasma membranes include specific channels for several common ions. Also, carrier proteins, that's another kind of integral protein. This type of protein transports a specific substance across the membrane by undergoing a change in shape. For example, amino acids needed to synthesize new proteins enter the body cells via carriers. Receptor proteins are an example of another integral protein. Receptor proteins recognize specific ligands and they can alter a cell's function in some way. For example, antidiuretic hormone binds to the receptors in the kidneys and changes the water permeability of certain plasma membranes. Some enzymes function as membrane proteins, and these type of proteins can be either integral or peripheral. Enzyme proteins catalyze reactions inside of the cell or outside of the cell. For example, lactase, protruding from the epithelial cells lining your small intestine, splits the disaccharide lactose in the milk you drink. Another type of membrane protein is the linker protein, and these proteins can also be integral or peripheral. Linker proteins anchor filaments inside and outside of the plasma membrane, and that provides structural stability and shape for the cell. They may also participate in movement of the cell or link two cells together. And then cell identity markers, and this is a type of glycoprotein. This distinguishes your cells from anyone else's. Moving on to membrane fluidity. Membranes are fluid structures, that is, most of the membrane lipids and many of the membrane proteins can easily rotate and move sideways in their own half of the bilayer. Neighboring lipid molecules exchange place about 10 million times per second and may wander completely around the cell in only a few minutes. Membrane fluidity depends on both the number of double bonds in the fatty acid tails of the lipids that make up the bilayer and on the amount of cholesterol present. Each double bond puts a quote-unquote kink in the fatty acid tail, which increases membrane fluidity by preventing lipid molecules from packing tightly in the membrane. Membrane fluidity is an excellent compromise for the cell. A rigid membrane would lack mobility, and a completely fluid membrane would lack structural organization and mechanical support that is required by the cell. Membrane fluidity allows the interactions to occur within the plasma membrane, such as the assembly of membrane proteins. It also enables the movement of the membrane components responsible for cellular processes such as cell movement, growth, division, and secretion, and the formation of cellular junctions. Fluidity allows the lipid bilayer to self-seal if torn or punctured. For example, when a needle is pushed through the plasma membrane and pulled out, the puncture site seals spontaneously and the cell does not burst. Moving on to membrane permeability, the term permeable means that a structure permits the passage of substances through it, while impermeable means a structure does not permit the passage of substances through it. The permeability of the plasma membrane to different substances varies. Plasma membrane permits some substances to pass more readily than others. This property of the plasma membrane is termed selective permeability. The lipid bilayer portion of the plasma membrane is highly permeable to nonpolar molecules such as oxygen, carbon dioxide, and steroids. The plasma membrane is moderately permeable to small, uncharged polar molecules such as water and urea. The plasma membrane is impermeable to ions and large, uncharged polar molecules such as glucose. Transmembrane proteins that act as channels and carriers increase the plasma membrane's permeability to a variety of ions and uncharged polar molecules that, unlike water and urea molecules, cannot cross the lipid bilayer unassisted. Channels and carriers are very selective. Each one helps a specific molecule or ion to cross the membrane. Macromolecules, such as proteins, are so large that they are unable to pass across the plasma membrane except by endocytosis and exocytosis. And that brings us to gradients across the plasma membrane. The selective permeability of the plasma membrane allows a living cell to maintain different concentrations of certain substances on either side of the plasma membrane. A concentration gradient is a difference in the concentration of a chemical from one place to another, such as from the inside of the cell to the outside of the plasma membrane. Many ions and molecules are more concentrated in either the cytosol or the extracellular fluid. For instance, oxygen molecules and sodium ions are more concentrated in the extracellular fluid than in the cytosol, and it is the opposite for carbon dioxide and potassium ions. The plasma membrane also creates a difference in the distribution of positively and negatively charged ions between the two sides of the plasma membrane. Typically, the inner surface of the plasma membrane is more negatively charged and the outer surface is more positively charged. A difference in electrical charges between the two regions constitutes an electrical gradient. Because it occurs across the plasma membrane, this charge difference is termed the membrane potential. Moving on to transport across the plasma membrane, substances generally move across the cellular membranes via transport processes that can be classified as passive or active. This depends on whether they require energy or not. In the passive process, a substance moves down its concentration or electrical gradient to cross the membrane using its own kinetic energy. Kinetic energy is intrinsic to the particles that are moving. This is because there is no input of energy from the cell. In the active processes, cellular energy is used to drive the substance uphill or against its concentration or electrical gradients. The cellular energy used is usually in the form of ATP. An example is active transport. Another way that some substances may enter the cell or leave the cell is an active process in which tiny spherical membrane sacs referred to as vesicles are used. Examples of this include endocytosis, which the vesicles detach from the plasma membrane while bringing materials into a cell, and exocytosis, which is the merging of the vesicles with the plasma membrane to release the material from the cell. Learning why materials diffuse across membranes requires an understanding of how diffusion occurs in a solution. Diffusion is a passive process in which the random mixing of particles in a solution occurs because of the particles' kinetic energy. Both the solutes, the dissolved substances, and the solvent, that is the liquid that does the dissolving, undergo diffusion. If a particular solute is present in high concentration in one area of a solution and low concentration in another area, the solute molecules will diffuse toward the area of lower concentration. In other words, they move down their concentration gradient. After some time, the particles become evenly distributed through the solution, and the solution is said to be at equilibrium. The particles continue to move about randomly due to their kinetic energy, but their concentrations do not change. So simple diffusion is a passive process in which substances move freely through the lipid bilayer of the plasma membranes of cells without help from the membrane transport proteins. So nonpolar, hydrophobic molecules move across the lipid bilayer through the process of simple diffusion. Such molecules include oxygen, carbon dioxide, nitrogen gases, fatty acids, steroids, and fat-soluble vitamins such as vitamin A, vitamin D, vitamin E, and vitamin K. Also, small uncharged polar molecules such as water, urea, and small alcohols also pass through the lipid bilayer by simple diffusion. Solutes that are too polar or highly charged to move through the lipid bilayer by simple diffusion can cross the plasma membrane by a passive process called facilitated diffusion. In this process, an integral membrane protein assists a specific substance across the membrane. The integral membrane proteins can either form a channel or function as a carrier. Osmosis is a type of diffusion in which there is a net movement of a solvent through a selective permeable membrane. Like other types of diffusion, osmosis is a passive process. In living systems, the solvent is water, which moves by osmosis across plasma membranes from an area of higher water concentration to an area of lower water concentration. Another way to understand this idea is to consider the solute concentration. In osmosis, water moves through a selective permeable membrane from an area of lower solute concentration to an area of higher solute concentration. During osmosis, water molecules pass through a plasma membrane in two ways. One, by moving between neighboring phospholipid molecules in the lipid bilayer via simple diffusion as previously described. Or two, by moving through aquaporins. Aquaporins are membrane proteins that function as water channels. In primary active transport, energy that is derived from the hydrolysis of ATP changes the shape of a carrier protein which quote-unquote pumps a substance across the plasma membrane against its concentration gradient. In secondary active transport, the energy stored in a sodium ion or a hydrogen ion concentration gradient is used to drive other substances across the membrane against their own concentration gradients. Because a sodium ion or a hydrogen ion gradient is established by the primary active transport, secondary active transport indirectly uses energy obtained from the hydrolysis of ATP. As noted earlier, a vesicle is a small spherical sac. Vesicles also import materials from and release materials into the extracellular fluid. During endocytosis, materials move into a cell in a vesicle formed from the plasma membrane. In exocytosis, materials move out of a cell by the fusion with the plasma membrane of vesicles formed inside of the cell. Both endocytosis and exocytosis require energy that is supplied by ATP. Thus, transport in vesicles is an active process. And this is figure 3.4 on page 69. So diffusion is influenced by the steepness of the concentration gradient. In other words, the greater the difference in the concentration between the two sides of the membrane, the higher the rate of diffusion. It can also be influenced by temperature. The higher the temperature, the faster the rate of diffusion. It can also be influenced by the mass of the diffusing substances. The larger the mass of the diffusing particle, the slower its diffusion rate. Smaller molecules diffuse more rapidly than larger ones. Also, the surface area can affect diffusion. The larger the membrane surface area available for diffusion, the faster the diffusion rate will occur. And also the distance of diffusion. The greater the distance over which the diffusion must occur, the longer it takes. Moving on to the next slide, transmembrane proteins help solutes that are too polar or too highly charged move through the lipid bilayer. The processes involved are channel-mediated facilitated diffusion. Most membrane channels are ion channels, and carrier-mediated facilitated diffusion. Carriers are transporters. So this cartoon is figure 3.5 on page 70. This cartoon outlines simple diffusion, channel-mediated facilitated diffusion, and carrier-mediated facilitated diffusion. In simple diffusion, a substance moves across the lipid bilayer of the plasma membrane without the help of membrane transport proteins. In facilitated diffusion, a substance moves across the lipid bilayer aided by a channel protein or a carrier protein. And this slide brings us to figure 3.6, also on page 70. This outlines channel-mediated facilitated diffusion of potassium ions through a gated potassium ion channel. A gated channel is one in which a portion of the channel proteins acts as a gate to open or close the channel's pores to the passage of ions. Channels are integral membrane proteins that allow the specific small inorganic ions to pass across the membrane by facilitated diffusion. That brings us to figure 3.7 on page 71. Figure 3.7 outlines carrier-mediated facilitated diffusion of glucose across a plasma membrane. So at step 1, glucose binds to a specific type of carrier protein called the glucose transporter on the outside of the surface of the membrane. And at step 2, as a transporter protein undergoes a change in shape, glucose passes through the membrane. And finally, at step 3, the transporter releases glucose on the other side of the membrane. And this cartoon is on page 72. This is figure 3.8. This cartoon outlines the principle of osmosis. Osmosis is the movement of water molecules through a selectively permeable membrane. The solutes cannot cross the selectively permeable membrane, but water can. And that brings us to tonicity. A solution's tonicity is a measure of the solute's ability to change the volume of cells by altering their water content. So looking at figure 3.9 on page 73, we can see that red blood cells placed in an isotonic solution maintain their shape because there is no net water movement into or out of the cells. On the other hand, in a hypotonic solution, that is a solution that has a lower concentration of solutes than the cytosol inside of the red blood cell, we see that the cell can undergo hemolysis. In other words, it can rupture. And in a hypertonic solution, there is a higher concentration of solutes in the solution than there is in the red blood cell. In this case, the cell undergoes crenation, or it shrivels. This table is also part of figure 3.9 on page 73, so I'll let you go through this table on your own time. And that brings us to figure 3.10 on page 74. This figure outlines the sodium-potassium pump that expels sodium ions and brings potassium ions into the cell. So at step 1, 3 sodium ions in the cytosol bind to the pump protein. At step 2, The binding of sodium ions triggers the hydrolysis of ATP into ADP, a reaction that also attaches a phosphate group to the pump protein. The chemical reaction changes the shape of the pump protein, expelling 3 sodium ions into the extracellular fluid. Now the shape of the pump protein favors the binding of 2 potassium ions in the extracellular fluid to the pump protein. At step 3, the binding of potassium ions triggers the release of the phosphate group from the pump protein. This reaction again causes the shape of the pump protein to change. And at step 4, as the pump protein reverts back to its original shape, it releases potassium ions into the cytosol. At this point, the pump is again ready to bind to 3 sodium ions, and then the cycle repeats. And this brings us to figure 311 on page 75. This outlines the secondary active transport mechanism. The secondary active transport mechanisms use the energy stored in an ionic concentration gradient. Because the primary active transport pumps that hydrolyze ATP maintain the gradient, the secondary active transport mechanisms consume ATP indirectly. So at letter A, you see that antiporters carry 2 substances across the membrane in opposite directions, and at B, symporters carry 2 substances across the membrane in the same direction. That brings us to figure 3.12 on page 76. So this cartoon outlines receptor-mediated endocytosis of a low-density lipoprotein particle. The first step is binding. So on the extracellular side of the plasma membrane, an LDL particle that contains cholesterol binds to a specific receptor in the plasma membrane to form a receptor LDL complex. At step 2, the vesicle forms. The invaginated edges of the membrane around the clathrin-coated pit fuse, and a small piece of the membrane pinches off. At the uncoating step, step 3, almost immediately after it is formed, the clathrin-coated vesicle loses its clathrin coat and becomes an uncoated vesicle. At step 4, where the fusion with the endosome occurs, the uncoated vesicle quickly fuses with the vesicle known as the endosome. And at step 5, the recycling of the receptors to the plasma membrane, most of the receptors accumulate in the elongated protrusions of the endosome. These pinch off, forming transport vesicles that return the receptors to the plasma membrane. And at step 6, the degradation in lysosomes, other transport vesicles, which contain LDL particles, bud off of the endosome and soon fuse with the lysosome. Lysosomes contain many digestive enzymes. Certain enzymes break down the large protein and lipid molecules of the LDL particle into amino acids, fatty acids, and cholesterol. Moving on to figure 3.13 on page 77, this cartoon outlines phagocytosis. Phagocytosis is a vital defense mechanism that helps protect the body from disease. And now we're looking at figure 3.14 on page 78. And this cartoon shows bulk phase endocytosis. This is where the plasma membrane folds inward, forming a vesicle. Most body cells carry out bulk phase endocytosis, the non-selective uptake of tiny droplets of extracellular fluid. Moving on to exocytosis and transcytosis. In contrast with endocytosis, which brings materials into the cell, exocytosis releases materials from a cell. All cells carry out exocytosis, but it is especially important in 2 types of cells. One is the secretory cells that liberate the digestive enzymes, hormones, mucus, or other secretions, and 2, the nerve cells that release substances called neurotransmitters. Transport in vesicles may also be used to successfully move a substance into, across, and out of a cell. In this active process called transcytosis, vesicles undergo endocytosis on one side of a cell, move across the cell, and then undergo exocytosis on the opposite side. And table 3.1 is on page 79 in your textbook. This is a comparison of transport types, so please review this table on your own time. This slide is also part of the same table, the same page. This slide is also part of the same table and the same page. This slide is also part of the same table and the same page. And this slide is also part of the same table and the same page. Moving on to the cytoplasm. The cytoplasm consists of all the cellular contents between the plasma membrane and the nucleus, and it has two components, the cytosol and the organelles. The cytosol is the fluid portion of the cytoplasm that surrounds the organelles and constitutes about 55% of the total cell volume. And the organelles are specialized structures that have specific shapes and perform specific functions in the cell. So this table 3.2 is found on pages 91 and 92 in your textbook. It outlines the cell parts and their functions. So please review this table on your own time. This is slide 2 of 6 of the same table, same pages. Slide 3 of 6. Slide 4 of 6. Slide 5 of 6. And slide 6 of 6. And that brings us to figure 3.15 on page 80. The cytoskeleton is a network of protein filaments that extends throughout the cytosol. 3 types of filaments contribute to the cytoskeleton's structure as well as the structure of other organelles. In the order of their increasing diameter, these structures are microfilaments, intermediate filaments, and microtubules. Microfilaments are the thinnest elements of the cytoskeleton. They are composed of proteins actin and myosin, and they are the most prevalent at the edge of the cell. Intermediate filaments are thicker than microfilaments, but thinner than microtubules. Several different proteins can compose intermediate filaments, which are exceptionally strong. And microtubules are the largest of the cytoskeleton components. They are long, unbranched, hollow tubes, mainly of the protein tubulin. And now we're looking at figure 3.16 on page 81. The centrosome is located near the nucleus and consists of 2 components, a pair of centrioles and the paracentriolar matrix. The 2 centrioles are cylindrical-shaped structures, each composed of 9 clusters of microtubule triplets arranged in a circular pattern. Surrounding the centrioles is the paracentriolar matrix. This contains hundreds of ring-shaped complexes composed of the protein tubulin. These tubulin complexes are organizing centers for the growth of the mitotic spindle, which plays a critical role in cell division. Also, the mitotic spindle plays a critical role in the development of the microtubules in non-dividing cells. Moving on to figure 3.17 on page 82. Microtubules are the dominant components of cilia and flagella. Cilia and flagella are motile projections of the cell surface. Cilia are numerous, short, hair-like projections that extend from the surface of the cell. Each cilium contains a core of 20 microtubules surrounded by a plasma membrane. Flagella are similar in structure to cilia, but are typically much longer, and in contrast, flagella usually move an entire cell. So this next slide is still figure 3.17, but it's showing the differences in movement between the cilia and the flagellum. And now we're looking at figure 3.18 on page 83. Ribosomes are the sites of protein synthesis. The name of these tiny structures reflects their high content of one ribonucleic acid, but each ribosome also includes more than 50 proteins. Structurally, a ribosome consists of two subunits, one about half the size of the other. The large and small subunits are made separately in the nucleolus. This is a spherical body inside of the nucleus, and once produced, the large and small subunits exit the nucleus separately, then come together in the cytoplasm. Moving on to the endoplasmic reticulum. This is figure 3.19 on page 84. The endoplasmic reticulum is a network of membranes in the form of flattened sacs or tubules. The endoplasmic reticulum, or ER, extends from the nuclear envelope to which it is connected and projects throughout the cytoplasm. The ER is so extensive that it constitutes more than half of the membranous surfaces within the cytoplasm of most cells. Furthermore, cells contain two distinct forms of ER, which differ in structure and function. Rough ER is continuous with the nuclear membrane and usually is folded into a series of flattened sacs. The outer surface of rough ER is studded with ribosomes. Proteins synthesized by ribosomes attached to the rough ER enter spaces within the ER for processing and sorting. Smooth ER extends from the rough ER to form a network of membrane tubules. Unlike rough ER, smooth ER does not have ribosomes on the outer surfaces of its membrane. However, smooth ER contains unique enzymes that make it functionally more diverse than rough ER. Because it lacks ribosomes, smooth ER does not synthesize proteins, but it does synthesize fatty acids and steroids such as estrogens and testosterone. In liver cells, enzymes of the smooth ER help release glucose into the bloodstream and inactivate or detoxify lipid-soluble drugs or potentially harmful substances such as alcohol, pesticides, and carcinogens. And this next slide is still figure 3.19 on page 84. And this slide is simply showing us scanning electron microscope images of smooth ER and rough ER. Moving on to the Golgi complex, this is figure 3.2 on page 85. Most of the proteins synthesized by the ribosomes attached to the rough ER are ultimately transported to other regions of the cell. The first step in the transport pathway is through the organelle called the Golgi apparatus. The Golgi complex consists of 3 to 20 saccules, small, flattened membranous sacs with bulging edges that resemble a stack of pita bread. The saccules are often curved, giving the Golgi complex a cup-like shape. Most cells have several Golgi complexes, and Golgi complexes are more extensive in cells that secrete proteins. Different enzymes in the entry and exit saccules of the Golgi complex permit each of these areas to modify, sort, and package proteins into vesicles for transport to different destinations. The entry face receives and modifies proteins produced by the rough ER. The intermediate saccules add carbohydrates to proteins to form glycoproteins and lipids to proteins to form lipoproteins. And the exit face modifies the molecules further and then sorts them and packages them for transport to their destinations. And we're now looking at the cartoon on page 86. This is figure 3.21. This cartoon outlines the processing and packaging of proteins by the Golgi complex. At step 1, proteins synthesized by ribosomes on the rough ER are surrounded by a piece of the ER membrane, which eventually buds from the membrane surface to form transport vesicles. At step 2, the transport vesicles move toward the entry face of the Golgi complex. Step 3, we see the fusion of several transport vesicles, creating the entry face of the Golgi complex and releasing proteins into its lumen. At step 4, the proteins move from the entry face into one or more intermediate saccules. enzymes in the medial saccules modify the proteins to form glycoproteins, glycolipids, and lipoproteins. Transfer vesicles that bud from the edges of the saccules move specific enzymes back toward the entry face and move some partially modified proteins towards the exit face. At step 5, the products of the intermediate saccules move into the lumen of the exit face. At step 6, within the exit face of the saccule, the products are further modified and are sorted and packaged. At step 7, we see some of the processed proteins that leave the exit face are stored in secretory vesicles. These vesicles deliver the proteins to the plasma membrane, where they are discharged by exocytosis into the extracellular fluid. For example, certain pancreatic cells release the hormone insulin this way. At step 8, other processed proteins leave the exit face in membrane vesicles that deliver their contents to the plasma membrane for incorporation into the membrane. In doing so, the Golgi complex adds new segments of plasma membrane as existing segments are lost and modifies the number and distribution of the membrane molecules. And at step 9, finally, some processed proteins leave the exit face in transport vesicles that will carry the proteins to another cellular destination. Moving on to lysosomes, figure 3.22 on page 87. Lysosomes are membrane-enclosed vesicles that form from the Golgi complex. They can contain as many as 60 kinds of powerful digestive and hydrolytic enzymes that can break down a wide variety of molecules once the lysosome fuses with the vesicles formed during endocytosis. Because lysosomal enzymes work best at an acidic pH, the lysosomal membrane includes active transport pumps that import hydrogen ions. Thus, the lysosomal interior has a pH of 5, which is 100 times more acidic than the pH of the cytosol. The pH of the cytosol should be pH 7. The lysosomal membrane also includes transporters that move the final products of digestion, such as glucose, fatty acids, and amino acids, into the cytosol. Moving on to the next slide, lysosomal enzymes help recycle worn-out cell structures. A lysosome can engulf another organelle, digest it, and return the digestive components to the cytosol for reuse. In this way, old organelles are continually replaced. The process by which the entire worn-out organelles are digested is called autophagy. In autophagy, the organelle to be digested is enclosed by a membrane derived from the ER to create a vesicle called an autophagosome. The vesicle then fuses with the lysosome. In this way, a human liver cell, for example, recycles about half of its cytoplasmic contents every week. Autophagy is also involved in cellular differentiation, control of cell growth, tissue remodeling, adaptation to adverse environments, and cell defense. Lysosomal enzymes may also destroy the entire cell that contains them. This is a process known as autolysis. Autolysis occurs in some pathological conditions and is also responsible for the tissue deterioration that occurs immediately after death. Moving on to peroxisomes, this is another group of organelle in similar structure to the lysosome, but peroxisomes are smaller. Peroxisomes, also called microbodies, contain several oxidases, that is, enzymes that can oxidize various organic substances. For instance, amino acids and fatty acids are oxidized in peroxisomes as part of the normal metabolism. In addition, enzymes in peroxisomes oxidize toxic substances such as alcohol. Moving on to proteasomes, the continuous destruction of unneeded, damaged, or faulty proteins is the function of these tiny barrel-shaped structures that consist of 4 stacked rings of proteins around a central core called a proteasome. And this figure is 3.23 on page 89. Because they generate most of the ATP through aerobic respiration, they are referred to as the powerhouses of the cell. These are the mitochondria. A cell may have as few as 100 or as many as several thousand mitochondria, depending on its activity. Active cells that use ATP at a high rate, such as those found in the muscles, liver, and kidneys, have a large number of mitochondria. Moving on to figure 3.24 on page 90, the nucleus is a spherical or oval-shaped structure that usually is the most prominent feature of a cell. Most cells have a single nucleus, although some, such as mature red blood cells, have none. In contrast, skeletal muscle fibers and a few other types of cells have multiple nuclei. A double membrane called the nuclear envelope separates the nucleus from the cytoplasm. Both layers of the nuclear envelope are bilayers similar to the plasma membrane. The outer layer of the nuclear envelope is continuous with the rough ER and resembles it in structure. Many openings called nuclear pores extend through the nuclear envelope. Each nuclear pore consists of a circular arrangement of proteins surrounding a large central opening that is about 10 times wider than the pore of a channel protein in the plasma membrane. Nuclear pores control the movement of substances between the nucleus and the cytoplasm. Moving on to the next slide, the nucleus contains the hereditary units of the cell called genes, and the genes are arranged along chromosomes. And this brings us to page 93, figure 3.26. This shows an overview of gene expression. So the synthesis of a specific protein requires the transcription of a gene's DNA into RNA and the translation of RNA into the corresponding sequence of amino acids. This is called the central dogma of molecular biology. Moving on to the next slide, transcription occurs in the nucleus. The genetic information represented by the sequence of base triplets in DNA serves as a template for copying the information into a complementary sequence of codons. 3 types of RNA are made from the DNA template. Those types of RNA are messenger RNA, ribosomal RNA, and transfer RNA. Moving on to the next slide, messenger RNA directs protein synthesis. Ribosomal RNA joins with ribosomal proteins to make ribosomes. And transfer RNA binds to an amino acid and holds it in place during translation. Moving on to the next slide, in the process of translation, the nucleotide sequence in an mRNA molecule specifies the amino acid sequence of a protein. Ribosomes in the cytoplasm carry out translation. The small subunit of a ribosome has a binding site for mRNA. The larger subunit has 3 binding sites for the tRNA molecules. We call that a P site, an A site, and an E site. And that is outlined for you in this cartoon on this slide, and this is figure 3.28 on page 94. Moving on to figure 3.29 on page 95, we see that during protein synthesis, the small and large ribosomal subunits join to form the functional ribosome. When the process is complete, they will separate. So at step 1, an mRNA molecule binds to the small ribosomal subunit at the mRNA binding site. Then a special tRNA, called an initiator tRNA, binds to the start codon, which is AUG on the mRNA. This is where translation begins. The tRNA anticodon, UAC, attaches to the mRNA codon, AUG, by pairing between the complementary bases. Besides being the start codon, AUG is also the codon for the amino acid methionine. Thus, methionine is always the first amino acid in a growing polypeptide. At step 2, the large ribosomal subunit attaches to the small ribosomal subunit. This is called the mRNA complex. This creates a functional ribosome. The initiator tRNA with its amino acid methionine fits into the P site of the ribosome. Then at step 3, the anticodon of another tRNA with its attached amino acid pairs with the second mRNA codon at the A site of the ribosome. At step 4, a component of the larger ribosomal subunit catalyzes the formation of a peptide bond between methionine and the amino acid carried by the tRNA at the A site. Then at step 5, following the formation of the peptide bond, the resulting 2-peptide protein becomes attached to the tRNA at the A site. At step 6, after the peptide bond formation, the ribosome shifts the mRNA strand by 1 codon. The tRNA in the P site enters the E site and is subsequently released from the ribosome. The tRNA in the A site, bearing the 2 peptide protein shifts into the P site. This allows another tRNA with its amino acid to bind to the newly exposed codon at the A site. Then, steps 3 through 6 occur repeatedly, and the protein lengthens progressively. Finally, protein synthesis ends when the ribosome reaches a stop codon at the A site, which causes the completed protein to detach from the final tRNA. In addition, tRNA vacates the P site and the ribosome splits into its large and small subunits. This is figure 3.31 on page 98, and this shows the replication of DNA. So the 2 strands of the double helix separate by the breaking of the hydrogen bonds. The hydrogen bonds are shown as the dashed lines between the nucleotides. New complementary nucleotides attach to the proper sites, and a new strand of DNA is synthesized alongside each of the original strands. Arrows indicate hydrogen bonds forming again between these pairs. So a couple things to notice here is that A always pairs with T and G always pairs with C. Also, there are 3 hydrogen bonds between G and C, and there are only 2 hydrogen bonds between A and T. So G and C create a stronger bond than A and T do. Moving on to cell division, this is figure 330 on page 97. Most cells of the human body undergo cell division. This is the process by which cells reproduce themselves. The 2 types of cell division is either somatic cell division or reproductive cell division, and they accomplish different goals for the organism. A somatic cell is any cell of the body other than a germ cell. A germ cell is a gamete, which means a sperm or an egg, or we could also call that a reproductive cell. In somatic cell division, a cell undergoes a nuclear division called mitosis and a cytoplasmic division called cytokinesis. And this will produce 2 genetically identical cells, each with the same number and kind of chromosomes as the original cell. Somatic cell division replaces dead or injured cells and new ones during tissue growth. On the other hand, reproductive cell division is the mechanism that produces the gametes. Gametes are the cells that are needed to form the next generation of sexually reproducing organisms. This process consists of a special 2-step division that's called meiosis, in which the number of chromosomes in the nucleus is reduced by half. So in this figure, we see that in a complete cell cycle, the starting cell duplicates its contents and divides into 2 identical cells. Moving on to interphase, during interphase, the cell replicates its DNA. It also produces additional organelles and cytosolic components in anticipation of cell division. Interphase is the state of high metabolic activity. It is during this time that the cell does most of its growing. Interphase consists of 3 phases, G1, S1, and G2. The G1 phase is the interval between the mitotic phase and the S phase. During G1, the cell is metabolically active. It replicates most of its organelles and cytosolic components, but not its DNA. The S phase is the interval between G1 and G2, and it lasts about 8 hours. During the S phase, DNA replication occurs, As a result of DNA replication, the two identical cells formed during cell division later in the cell cycle will have the same genetic material. G2 is the phase between the S phase and the mitotic phase, and it lasts for about 4 to 6 hours. During G2, cell growth continues, enzymes and other proteins are synthesized in preparation for cell division. Moving on to the next slide. The mitotic phase of the cell cycle, which results in the formation of two identical cells, consists of a nuclear division, which we call mitosis, and a cytoplasmic division called cytokinesis, and this forms the two identical cells. The events that occur during mitosis and cytokinesis are plainly visible under a microscope because chromatin condenses into discrete chromosomes. Mitosis is a distribution of two sets of chromosomes into two separate nuclei. The process results in the exact partitioning of genetic information. For convenience, biologists divide the process into 4 stages: prophase, metaphase, anaphase, and telophase. However, mitosis is a continuous process. One stage merges seamlessly into the next. In prophase, the chromatin fibers condense and shorten into chromosomes that are visible under a light microscope. Because longitudinal DNA replication took place during the S phase of interphase, each prophase chromosome consists of a pair of identical strands called sister chromatids. A constricted region called a centromere holds the chromatid pair together. At the outside of each centromere is a protein complex known as a kinetochore. Later in prophase, the centrosomes start to form the mitotic spindle. This is a football-shaped assembly of microtubules that attach to the kinetochore. As the microtubules lengthen, they push the centromeres to the poles, or the ends of the cell, so that the spindle extends from pole to pole. The mitotic spindle is responsible for the separation of the chromatids to opposite poles of the cell. Then the nucleolus disappears and the nuclear envelope breaks down. During metaphase, the microtubules of the mitotic spindle align the centromeres of the chromatid pairs at the exact center of the mitotic spindle. This plane of alignment of the centromeres is called the metaphase plate. During anaphase, the centromeres split, separating the two members of each chromatid pair, which move toward opposite poles of the cell. Once separated, the chromatids are termed chromosomes, and the chromosomes are pulled by the microtubules of the mitotic spindle during anaphase. They appear V-shaped because the centromeres lead the way, dragging the trail arms of the chromosomes toward the pole. The final stage of mitosis is telophase. Telophase begins after chromosomal movement stops. The identical sets of chromosomes, now at opposite poles of the cell, uncoil and revert to the thread-like chromatin form. A nuclear envelope forms around each chromatin mass. The nucleoli reappear in the identical nuclei, and the mitotic spindle breaks up. And that brings us to cytokinesis. The division of a cell's cytoplasm and organelles into two identical cells is called cytokinesis. This process usually begins in late anaphase with the formation of a cleavage furrow. This is a slight indentation of the plasma membrane and is completed after telophase. The cleavage furrow usually appears midway between the centrosomes and extends around the periphery of the cell. Actin microfilaments that lie just inside the plasma membrane form a contractile ring that pulls the plasma membrane progressively inward. The ring constricts the center of the cell, like tightening a belt around a waist, and ultimately pinches it in two. Because the plane of the cleavage furrow is always perpendicular to the mitotic spindle, the two sets of chromosomes end up in separate cells. When cytokinesis is complete, interphase begins. So this brings us to table 3.3 on page 100. Please review this table on your own time. And this table is also on the same page. Please review it on your own time. Moving on to the control of cell destiny, there are 3 possible destinies for a cell. 1, it will remain alive and functioning without dividing. 2, it can grow and divide. Or 3, it can die. Homeostasis is when there's a balance between cell proliferation and cell death. Moving on to reproductive cell division. In the process called sexual reproduction, each new organism is the result of the union of 2 different gametes, one from each parent. If gametes had the same number of chromosomes as somatic cells, the number of chromosomes would double at fertilization. Meiosis is the reproductive cell division that occurs in the gonads. This produces gametes in which the number of chromosomes is reduced by half. As a result, the gametes contain a single set of 23 chromosomes and thus are termed haploid. Fertilization restores a diploid number of chromosomes. This cartoon is figure 3.33 on page 102. So unlike mitosis, which is complete after a single round of cell division, meiosis occurs in 2 successive stages, meiosis 1 and meiosis 2. During the interphase that precedes meiosis 1, the chromosomes of the diploid cell start to replicate. As a result of replication, each chromosome consists of 2 sister chromatids, which are attached to the centromeres. This replication of chromosomes is similar to the one that precedes mitosis in somatic cell division. Meiosis 1 begins once chromosomal replication is complete, and it consists of 4 phases: prophase 1, metaphase 1, anaphase 1, and telophase 1. Prophase 1 is an extended phase in which the chromosomes shorten and thicken and the nuclear envelope and nucleoli disappear and the mitotic spindle forms. 2 events that are not seen in mitotic prophase occur during prophase 1 of meiosis. First, the 2 sister chromatids of each pair of homologous chromosomes pair off in an event called synapsis. The resulting 4 chromatids form a structure called a tetrad. Second, parts of the chromatids of 2 homologous chromosomes may be exchanged with one another. Such an exchange between parts of non-sister chromatids is called crossing over. This process, among others, permits an exchange of genes between the chromatids of homologous chromosomes. Due to crossing over, the resulting cells are genetically unlike each other and genetically unlike the starting cell that produced them. Crossing over results in genetic recombination, that is, the formation of new combinations of genes. And this accounts for part of the great genetic variation among humans and other organisms that form gametes via meiosis. In metaphase 1, the tetrads formed by the homologous pairs of chromosomes line up along the metaphase plate of the cell with homologous chromosomes side by side. During anaphase 1, the members of each homologous pair of chromosomes separate as they are pulled to opposite poles of the cell by the microtubules attached to the centromeres. The paired chromatids, held together by a centromere, remain together. Telophase 1 and cytokinesis of meiosis are similar to telophase and cytokinesis of mitosis. The net effect of meiosis 1 is that each resulting cell contains the haploid number of chromosomes because it contains only one member of each pair of the homologous chromosomes present in the starting cell. And this next slide is also figure 3.33 on page 102. The second stage of meiosis is meiosis 2, and this also consists of 4 phases: prophase 2, metaphase 2, anaphase 2, and telophase 2. These phases are similar to those that occur during mitosis. The centromeres split, and the centrochromatids separate and move toward opposite poles of the cell. And that brings us to figure 3.34 on page 103. In summary, meiosis 1 begins with a diploid starting structure and ends with 2 cells, each with the haploid number of chromosomes. During meiosis 2, each of the 2 haploid cells formed during meiosis 1 divides. The The net result is 4 haploid gametes that are genetically different from the original diploid starting cell. Moving on to cellular diversity, cells vary considerably in size. The size of cells are measured in units called micrometers. The shapes of cells also vary considerably. They may be round, oval, flat, cube-shaped, column-shaped, elongated, star-shaped, cylindrical, or disc-shaped. A cell's shape is related to its function in the body. So of the nearly 100 trillion cells in the average adult human body, these cells can be classified into about 200 different cell types. Moving on to aging in cells, aging is a normal process accompanied by the progressive alteration of the body's homeostatic adaptive responses. It produces observable changes in the structure and function, and it increases the vulnerability to environmental stress and disease. The specialized branch of medicine that deals with the medical problems and care of the elderly person is called geriatrics. Moving on to disorders and homeostatic imbalances, cancer is a group of diseases characterized by uncontrolled or abnormal cell division. Therefore, cancer is a disease of the cell cycle. When the cells in a part of the body divide without control, the excessive tissue that develops is called a tumor. The study of tumors is called oncology. Tumors may be cancerous and often fatal, or they may be harmless. A cancerous neoplasm is called a malignant tumor, or a malignancy. One property of most malignant tumors is their ability to undergo metastasis. That is the spread of the cancerous cells to other parts of the body. A benign tumor is a neoplasm that does not metastasize. An example is a wart. Most benign tumors may be removed surgically if they interfere with normal body function or become disfiguring. Some benign tumors can be inoperable and perhaps fatal. The name of a cancer is derived from the type of tissue in which it develops. Most human cancers are carcinomas. These are malignant tumors that arise from epithelial cells. Melanoma, for example, are cancerous growths of melanocytes. These are skin epithelial cells that produce the pigment melanin. A sarcoma is a general term for any cancer arising from muscle fibers or connective tissues. For example, osteogenic sarcoma, the most frequent type of childhood cancer, destroys normal bone tissue. Leukemia is a cancer of blood-forming organs characterized by the rapid growth of abnormal leukocytes. And lymphoma is a malignant disease of the lymphatic tissue, for example, of the lymph nodes. Cells of malignant tumors duplicate rapidly and continuously as malignant cells invade the surrounding tissues. They often trigger angiogenesis. This is the growth of a new network of blood vessels. Proteins that stimulate angiogenesis in tumors are called tumor angiogenesis factors. The formation of new blood vessels can occur by either overproduction of tumor angiogenesis factors or by the lack of naturally occurring angiogenesis inhibitors. Then, as the cancer grows, it begins to compete with normal tissues for space and nutrients. Eventually, the normal tissue decreases in size and dies. Submalignant cells may detach from the initial tumor and invade the body cavity or enter the blood plasma or lymph plasma, then circulate and invade other body tissues, establishing secondary tumors. Malignant cells resist the anti-tumor defenses of the body. The pain associated with cancer develops when the tumor presses on nerves or blocks a passageway in an organ so that secretions build up pressure. Or the pain can also be a result of dying tissue or dying organs. So moving on to the next slide, several factors may trigger a normal cell to lose control and become cancerous. One cause is a carcinogen. Carcinogens induce mutations. These are permanent changes in the DNA base sequence of a gene. So a chemical agent, such as cigarette smoking, or radiation, as in UV rays, can produce cancer, and in this case, this is termed carcinogen. Oncogenes are cancer-causing genes. Most oncogenes derive from normal genes called proto-oncogenes that regulate growth and development. The proto-oncogene undergoes some change that causes it, 1, to be expressed inappropriately, 2, to make its products in excessive amounts, or 3, to make its products at the wrong time. Some cancers have a viral origin. Viruses are tiny packages of nucleic acids, either RNA or DNA, that can reproduce only while inside of the cells they infect. Some viruses, termed oncogenic viruses, cause cancer by stimulating the abnormal proliferation of cells. For instance, the human papilloma virus, HPV, causes virtually all cervical cancers in women. The virus produces a protein that causes proteasomes to destroy p53. p53 is a protein that normally suppresses unregulated cell division. In absence of this suppressor protein, cells proliferate without control. Traditional cancer treatment involves surgery, chemotherapy, and radiation. Many cancers are removed surgically. However, when cancer is widely distributed throughout the body or exists in organs such as the brain, whose functioning would be greatly harmed by surgery, chemotherapy and radiation therapy may be used instead. And there are other cancer therapies, such as immunotherapy. This is the use of various procedures that modify and stimulate the immune responses to fight cancer. Targeted therapy, which is the use of drugs that target specific genes and proteins found in cancer cells to stop cancer cells from growing and dividing or cutting off their blood supply. Stem cell transplants. This is a transplantation of stem cells to restore blood-forming cells in people whose red bone marrow has been destroyed by high doses of radiation and chemotherapy used to treat cancer. Also, hormone therapy is where using various levels of hormones to slow or stop the growth of cancer cells and tissues that are sensitive to hormonal growth control, such as breasts and the prostate. And complementary and alternative medicine therapy uses the remedies that lie outside of the traditional medical procedures, such as herbal, vitamin, and nutritional supplements, exercise, relaxation techniques, massage, prayer, hypnotherapy, and acupuncture. And that concludes tonight's lecture.